This is TWIS. This Week in Science, episode number 836, recorded on Wednesday, August 4th, 2021. Do you give a hoot about science? Hey there, I'm Dr. Kiki, and tonight on the show, we are going to fill your head with microbes, trade-offs, and melting. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. The following program depicts actual things being studied in the actual world by actual scientists. While the rest of the world engages in misinformation, either for entertainment or human manipulation purposes, we offer you a conversation about science. Not a class, not a lecture, nothing you will be expected to remember, just a conversation based in reality. A place we can all imagine humans will one day live, despite all the evidence to the contrary. If you haven't been here before, welcome. If you have, welcome back. It's time for This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki, and that's right, Blair's not here yet.
Yes, good science to you too, Justin and Blair, who will be joining us later, apparently. She's got other places to be tonight, but we're here to podcast, to talk about science. We've got all the science stories from the last week here to discuss with you because, yeah, as Justin said, this is a fun science conversation. So let's get talking. I brought stories. What did I bring tonight? I have stories related to some of those microbes. I also have a sinkhole, red bodies, and parenthood. What did you bring, Justin?
I also have a microbe story, and I have a segment that I think I introduced once or maybe twice, but then forgot about. It's the "Did we really need a study for that?" segment.
I always like those segments. It's like, yeah, really? Okay.
So this is the
common
thing about
sense,
the
but
segment.
let's back it up with science.
So the thing is, the segment is, did we really? And then sometimes it's like, probably not. And then sometimes it's like, oh yeah, maybe we did. Right? So it's you never know. It's a mixed bag segment. Also, something about a mass extinction on planet Earth and how Russia is melting.
Oh no, it's melting. Did somebody throw a bucket of water on the Witch of the West? Sorry. And I would ask Blair what's in the animal corner, but she's not here. So I'll just let you know that there's some legs coming, some long legs that will be coming up in that animal corner, among other science newsy bits related to animals. So let's jump on into the show. And as we do, I want to remind you all that if you have not yet subscribed to This Week in Science, you can find us. Look everywhere for This Week in Science TWIS. We are on YouTube, Facebook, and Twitch, streaming weekly the live broadcast of the show. And our podcast is available all over. We're TWIS Science on Instagram and on Twitter. And you can find our website at twis.org. Okay. It's time for the science. Let's dump bum 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 bum.
I already have theme music. You don't have to keep making up
right.
sounds.
Gotta. Sorry. Just do the theme music things every once in a while. It just keeps me going. Sound effects. Who brought the sound effect, Kiki? I brought some competitive microbes to start off the show. Did you ever think that it could have been the competition between microbes and also maybe the length of the day that was related to the oxygen in our atmosphere? Did that ever cross your mind previously?
No.
No, yeah, well, it crossed the minds of a few researchers who have been studying the question of where did all the oxygen come from in the Earth's atmosphere. We know there were a couple of oxygenation events throughout our multi-billion year history, right? So these events, many of them were related to cyanobacteria, blue-green algae that they grew within the waters of the planets, soaked up the energy of the sun, releasing oxygen as a byproduct of their metabolism. But were they just there? And how did it just come to happen that suddenly they put enough oxygen into the atmosphere so that there was enough oxygen for organisms that use oxygen as a source of energy to start living and surviving on the surface of the Earth? These researchers were looking at a sinkhole in Lake Huron. So this sinkhole at the bottom of Lake Huron is covered, the bottom of it is covered with bacterial microbial mats. And there are a few different species of microbes down there. The dominant species, however, are sulfur eating, so they don't use sunlight at all, and blue-green algae, cyanobacteria, that use the sun. And what seems to happen as these bacteria are existing on the bottom is that at night, when the sun goes away, the sulfur-eating bacteria start covering up the cyanobacteria. They grow, and because they are using all the energy of the sulfur that's bubbling up from the ground beneath the sinkhole as their energy source. They're dividing and multiplying and growing, and they're taking over until the sun comes out. And then the sunlight-using bacteria start growing and releasing oxygen, and there's lots of oxygen that gets released. And so these researchers discovered this, and they've been looking at the bottom of this deep sinkhole because it's this wonderful model system for what they think the early Earth might have been like. And they also thought back, oh my gosh, the Earth was spinning a lot faster once upon a time. So there was less daylight necessarily. The length of the day was like 6 hours at a time. It was a completely,
This?
billions of years ago.
Oh my gosh.
Yeah, so day length was completely different, much shorter.
I'm just trying to get this because this is very important. So like a work day would be like max 4 hours.
Yeah, like Matt, 2 hours. Come on.
Okay.
Yeah, and so they would have their sunlight work day for the cyanobacteria way back when, and it perhaps took a long period of time for these cyanobacteria to get enough oxygen or to get enough sunlight so that they could begin to grow and take over the extremophiles or the chemophiles who had been existing off of the sulfur as their energy source. And eventually, the Earth started slowing down and the day length became longer, more consistent, and we had more sunlight and oxygenation events. So maybe it was the spinning of the Earth in its orbit around the sun and how that changed through the geologic history of the planet that set the stage for the potential of the great oxygenation events to even occur. Yeah. A very, very fun combination of things. Not just the bacteria at play, but also how much sun was available and when that sun was available and where. Looks to me like we have a Blair. She has joined us. Hello. You made it.
Yeah. So happy to join.
Yeah, we are so glappy. We're glappy?
Are you glappy?
We're so glappy to have you here tonight to bring us the Animal Corner. We were just finishing up our first story. It's time for Justin to tell a story. What you got there, Justin?
So last week we talked a little about bees getting trained by caffeine to be better pollinators. But what about the flowers? How do we get flowers themselves to be better pollen producers? One way we could support the plant's natural abilities has been discovered by Sean Christensen of the UC Davis Department of Entomology and Nematology in the wonderful town of Davis, California. Christensen, quoting here, "We found out that certain bacteria in flowers, Acinetobacteria or Bacter, can send a chemical signal to pollen that hijacks its systems and tells it to open the door from the inside, releasing protein and nutrients for the bacteria. In terms of potential significance, this bacteria can double the amount of protein released from pollen. So it's important for bacterial growth, but it also could be exploited by bees or other pollen consumers to get more nutrition from their food. In the study, they found that the bacteria induced over 5 times greater pollen germination and 20 times greater pollen bursting than that of uninoculated pollen. There was also, they had found some nectar-inhabiting yeast that they studied that neither produced nor benefited anything, had nothing to do with pollen germination. So they conclude that Acinetobacter both specifically causes and benefits from inducing pollen germination and burst. So now that we have this whole other thing we can look at in farming in terms of what's on what bacteria is present and in what amounts, and should we be seeding? Should we be spraying bacteria on plants instead of insecticides to get more of these pollinators attracted to come and do their job?
Yeah, I mean, I want to know can we not just, how can we stimulate even more pollination when we have a decrease in pollinators? So if insects are dying, can we enhance the ability of flowers to find the pollinators they need?
Yeah, can we feed them? Can we get the plants that they are still being attracted to feed them better? And if so, should have stronger, healthier insect pollinators that are going around and pollinating more and keeping. This is one of those things too. It's like, who cares about a bacteria and a bee? Actually, our entire food web depends on them. Our entire food web is so critically dependent on pollinators in the state of California, which is the largest ag state in the United States. One of the largest food producing regions in the world. Absolutely critical that we do everything possible.
Yeah,
sure
so
it's intact.
It's good bacteria. The bacteria need to be there to protect, I guess, to also fight off fungi that might be bad for the flowers. The bacteria are going to
inhibitors. They can actually be pollination inhibitors. So having a bacteria there to fight off that fungal is absolutely, I think. And this is not something like weird out that's being added into nature. It's already present and they're finding it in nature. This is actually a sort of a microbiota of bees, this bacteria. It's because they're attracted to pollinators. This thing is on the pollen, so it's already in that system. But the idea is, you could actually enhance output of that pollen.
It's good for the flowers, good for the bacteria, good for the bees.
Good people.
Good for people. We are all, it is microbial earth, right? It's just the microbial web is what is important. Blair, are you ready to give a short story to us?
Oh, yeah, I am. Out, actually.
Oh, yeah.
I want to tell you about Daddy Shortlegs.
What? Okay. There's I know that I've heard of a Daddy Longleg. What's a Daddy Shortleg?
It's something scientists created. So anyway,
it's not Halloween yet. Hold on.
Yeah. Scientists shortened the legs of Daddy Longlegs and changed their functionality by tweaking their DNA expression. There was a reason for this, I will say, and it was to figure out which genes cause spider relatives to develop these crazy long legs. Makes them a daddy long legs. And the researchers assembled a draft genome of them, Phalangium opilio. They looked at 3 genes that act as a blueprint for where various body parts should go. And then they used an RNA interface, a technique that reduces gene expression to knock down hundreds of developing embryos, these specific genes. When they did so, 6 of 8 legs were half their normal size. And they had transformed into short pedipalps, which are like limbs for food handling. And when the researchers knocked down a third developmental gene that they thought was to help build embryonic legs, they didn't turn into pedipalps, but they did get shorter. And they also lost their tarsomeres. What's a tarsomere, you ask? I'm so glad you asked. Because it is a set of about 100 knuckle-like joints that can wrap around sticks, quote unquote, like a monkey's tail. So, basically, they helped identify the genes or the gene groups that select for the use and the type of legs that they have and leg length. So this is also similar to other studies that have been done in fruit flies. And so this gives scientists an idea for how these crazy long legs evolved, if they know kind of the gene that it came from. So it maybe is interesting, but this is a good reminder of just because you could doesn't mean you should.
But it's also short legs. But they're using RNA to suppress a gene that in development. Like this is like real time gene. This isn't. They didn't go in and edit the genes of the parents or the embryo and then have a next generation. They figured out which RNA can inhibit or stop the translation of an activity on the like. That's the really to me amazing part of this is that that's a thing now we can do real time. And if you go forward, take this spider study now and zip us into the future, that means they could actually potentially apply RNA to defeat, say, a birth defect that they can tell is coming by modulating how that gene expression takes place. Oh, wow.
Yeah. And I think that's an important point is, looking at these genes, which are responsible for body form and which go back to our, the sponges, our last universal common ancestor, right? Which ones split off and led to spiders and their eight limbs, and which genes are responsible for humans and our fingers and our toes, and how can we potentially use RNA in the future to control these things? How does RNA control the transcription and translation of this now?
Aaron Laura in the chat is saying, does this mean that we can make everyone tall now? No, Aaron
nope.
So what science, the plan, I'll just give it up. This plan that science has for the future with the exploding populations and too many people with too few resources is actually, we're going to make everyone much shorter. Mm-hmm. We're going to make everyone a maximum of 3 feet tall.
That makes no sense. No one will be able to reach the cabinet above the refrigerator, Justin.
That's because we'll make the cabinet smaller, and now your single story 1-bedroom apartment is a 2-story, maybe 3-bedroom, right? So
what about the Olympics, Justin?
Right.
They'll still take place.
The Olympics.
They'll just have a height limit. You must be under this height to compete.
Yes. And meanwhile, Gaurav wants to know, what about the daddy partially long legs, or as I like to call them, the daddy medium legs? You could also get that done as well. But yes, this is a terrifying in thought exercise, very interesting in actual scientific implications.
Okay. And as we're ending this story, I just want to point out I had no idea daddy longlegs could wrap their little legs around things like monkey paws or like a monkey tail.
Pretty wild.
This Sweet to me is mind-blowing.
Yes. It's gross.
No clue.
Listen, I am both spider averse and a spider advocate. So I will be the first.
Daddy longlegs are the best. I love daddy longlegs. Yeah. Come on.
But also
they're great.
Creepy.
No, they're fantastic. No, daddy longlegs are cool. But now I'm going to be like, ooh, daddy monkey legs. This is very different.
Ooh, long monkey daddy long monkey legs.
I don't know if that's better.
No?
I have no idea.
Trending tomorrow? No?
Alrighty, let's move on to another story. Speaking of legs, no, we're not talking about legs anymore. I'm going to talk about some the discovery of some very red bodies, not here on Earth, but in the asteroid belt, where they shouldn't have been, because apparently objects with a reddish tinge or a reddish reflectance should be trans-Neptunian objects. So we have these delineations in our solar system related to where objects were when they were created. And the distance from the sun determines what stuff freezes. And so there are different places, like there's the snow line for water. It's where all the water freezes. There's the snow line for carbon dioxide. There's the snow line for other organics to freeze as it gets further and further and further away from the sun. And red bodies are typically found out past Neptune. They formed super far away from the sun. Everything's frozen on them. Yet these objects, these two objects that have been discovered, 203 Pompeia and 269 Justitia, are super red, and they're basically somewhere that researchers did not expect to find them. So what they can think based on this, the implications are that when Jupiter moved at some point during the development of our solar system, it knocked things around. And the gravitational pull and gravitational forces of these movements of these big planets potentially jostled some of these trans-Neptunian objects and made them come in closer to Earth, where they have taken up residence in the asteroid belt. And within the asteroid belt, this makes them a tantalizing target for exploring these objects to get evidence about the earliest stages of our solar system, but without having to go all the way out past Neptune. So we can explore some of these frozen organics. We can get samples potentially, like we've gotten before, from other asteroids and bring them back and potentially learn a lot about the early solar system and some of these objects that got stuck in an early developmental phase of the solar system. So, potentially easier targets to go out and learn more about our solar system without having to travel as far away from home. So it would be less time, less money intensive, and we could learn a lot. So, very red bodies. Researchers are excited about them.
That's a clever find.
Yeah. You know, and where there are two, there are probably more. So now that researchers are looking for this kind of a signature, potentially we will find more out in the asteroid belt as we continue looking. What else do you have, Justin?
Okay, this is the occasional reoccurring segment called Did We Really Need a Study for That? Study from the University of Kansas, published in the journal Appetite, finds food choices at all-you-can-eat buffet tied to weight gain.
What?
So, well, the immediate takeaway on the story is that
If you eat all the French fries, then
choosing to eat at an all-you-can-eat buffet is likely tied to weight gain, regardless of the choices you make after entering. Like that's choosing to study people in an all-you-can-eat environment is likely to give you a view of people who aren't also making the wisest food choices. But the study actually has some nuance to it. The researchers took young participants without obesity to an all-you-can-eat buffet, recorded their food choices. The researchers measured their body composition before they went there. A year later, they measured body composition again, which you would think, but wait, have they been at the buffet for the whole year?
Yeah, every day.
No, they just went once, but they recorded what foods they were picking out. And then found correlations between what types of food they were putting on their plate during that one-day visit and their body composition outcomes a year later. Participants who consumed a greater portion or proportion of carbohydrate foods and sodium, everything had sodium. So the sodium just sort of becomes noise. It's everything had sodium, like on pretty high levels. But the participants who consumed greater proportion of carbohydrate food in their buffet meals had significantly greater weight change and percent body fat change at the assessment a year later. The study found no significant body changes a year later for those in the buffet who ate high proportions of fat, high energy dense, and ultra processed foods. Researchers believe that there's what they have they're looking at here isn't necessarily something specific to the caloric quantitation of those foods that they were picking. Researchers believe that the weight gainers were choosing foods that triggered more of a reward system than the not weight gaining group, whose choices were more energy based. They had more of a physiological hunger satisfaction based on their foods than the group who was picking the carbohydrate foods that tended to be more of a psychological reward. An important factor that researchers also point out, so then that becomes the trend. What are you seeking food for? Like, I need to get a Cliff Bar. I need some protein. I need some energy to get through the day versus, I need to get like a glazed cinnamon bun thing because that's really what I'm craving. Like that's maybe the difference. One of the important factors that the researchers point out is that 90% of babies begin eating meals from adult plates, creating what they're calling sort of a cycle of
a feedback loop.
So what they're talking about also is hyper palatable foods. So these are foods with like lots of sugars or sodiums, things that are designed to trigger reward system. No manufactured baby food in the United States is in that hyper palatable category that would trigger that. However, if the first thing they're eating beyond that is something that is designed to get an adult human palate triggered, and reward system triggered, which probably has been an increase over time of how insane that can be. This is quoting Tara Fazzino, assistant professor of psychology at KU. One important, hyper palatable foods have combinations of ingredients that can enhance a food's palatability and make foods' rewarding properties artificially strong. If babies are consuming foods that are artificially higher rewarding at early in infancy, this could essentially indicate that there's to their system physiologically and to their brain that, hey, this is what food is supposed to taste like, and this is how rewarding it's supposed to be.
Okay. But is that what they studied? She's like making this assumption. This is not what the study was about. This is babies, they're going to be learning this from the grown-ups. Of course they're learning this from the grown-ups, but she didn't do that in the study. She just was like, hey, people make bad food choices, and this relates to their okay.
So my articulation. No, you're absolutely right. I 100% agree with you. That's a previous study that she did.
Okay.
So I
didn't articulate that based on a previous study on... Yes. You're absolutely right, but she's covered because she did that. She has done that.
Okay,
she has done that research. Yes,
I have one other thing I just want to mention is that you talked about body composition. Mm-hmm. So, is that BMI? Is that what that's about? Because BMI is like really problematic.
Yeah. I don't know. I don't think.
So I'm just curious.
I don't know what all of the factors that they looked at for assessing.
Because categorizing obesity, especially in children, is pretty hard.
Hang on. It was the other one was about what children are eating.
Right.
Yes.
But like the older study, right? Like when are they getting it?
But for this newer one, they were
young adults.
Body composition.
The newer one is young adults.
Earlier and then later. And
a year later.
Right.
Of young adults.
But I'm just
somewhere
saying,
in the 20s, I guess, is young adult.
Body composition is... I'm just putting out a word of caution that the whole... If they really are talking about BMI, which is what it sounds like, the whole foundation of BMI is very
they would have said BMI. So body composition, I don't know. I didn't look, but we could be talking about cholesterol levels. We could be talking about weight and height.
Body composition isn't cholesterol levels, but if they're doing a study, perhaps they are actually measuring total body fat because you can measure body fat to body muscle composition and actually get at composition. And that is going to be different than just
that
BMI.
ridiculous
Right.
thing where you stand on the thing and hold the two metal, like the electrical current tells you?
Yes.
Yeah.
So one of the ways that you also measure body fat percentage, which is also something that's recently been kind of called into question, is the little calipers, where you go and you pinch, and they say, okay, you're this fat based on just pinching a piece of your body. So it's I'm not saying that this is all bunk. I'm saying that when we're talking about human obesity, there are lots of different terms for what that means and lots of different ways to measure that. And some of them are used historically with very little reason.
Okay.
And that's all I'm saying. I'm not saying anything about this particular study. I just wanted to throw that out there.
According to the Google, body composition refers to the portion of fat you have relative to lean tissue. Muscles,
and they
bones,
looked
body,
at
water,
the percent of body fat change and weight change during the period of the study after 1 year.
I'm sorry, Blair, it's carbs.
What they ate at this ad libitum
There's no
meal.
defending them.
Listen, I'm not saying the carbs are not bad. I'm just saying, especially if you're talking about adolescents too. Like
these are in their 20s. Are those still adolescents?
Yeah, that's what you said before. You said adolescents. But anyway.
I did. Young adults. Yeah. I said young adults.
Oh, young adults. Okay.
Which is also a magnificent band. If you've never listened to the Young Adults, you should go find it.
Anyway, my point stands potentially even more for that group because the shape and distribution of weight changes in that group very rapidly. And I just wanted to throw it out there as food for thought. I don't need to have a whole
But no, she's a psychologist studying food. It's a food for thought. It's food for thought. That's exactly what the study was covering and with the results that they found.
I do think that what they're trying to look at is the pattern. So it's going to a buffet and how do people respond to a buffet? Do they respond with this, what they're calling hedonic eating, where it's like this, I am fulfilling a psychological need, the reward aspect, which what they're suggesting could go on and lead to a problem because then you're not watching what you eat as carefully as you should. You're just rewarding yourself at any chance you get. So that will lead to potential weight gain versus going in and making choices, actually picking and choosing and not just going for I want to feel really good from the carbs.
Yeah,
which is what I do. I go to a buffet and I'm like, give me all the rolls.
No, wow. See, I'm the opposite. I'm like, I need
salad.
I need to fatten up for winter off of this. I need to go get all the roast beef and bacon and put it on my plate.
Everybody has different ways, but I think it's an interesting way to get at this question of how do people respond to food and how does that potentially influence future weight gain?
And how are we trained on it?
And how are we trained on it? What is are you like a rat responding to a little lever that you have to press? I will eat the carbs, which I will. Give me the pie, the bread rolls. I'm going to eat them. Oh yeah, and the butter. And give me a whole thing of butter. I like that too. Okay, but everything in life is trade-offs. It's always got a trade-off. There's always a trade-off there. Well, some researchers just published their study in Nature Communications related to their work on a little molecule called CAM-K2. CAM kinase 2. This is calcium motor protein. And it's involved in a lot of muscle. And these researchers at Johns Hopkins Medicine have determined from their work with mice and fruit flies that this is a molecule of trade-off, that it gives that young vertebrates have evolved to have this CAM kinase in place for fight or flight, to go chase after prey, to run away from predators, to survive and be a success in life. But because CAM kinase is an oxidatively reductive molecule. It causes oxidation reactions. These oxidation reactions are what lead to many diseases of aging as we grow older. And so what they're thinking is that CAM kinase 2 may be, or at least the oxidation effects of CAM kinase 2 may be targets for therapy as we grow older, because this has the elevated CAM kinase activity has been linked to tissue damage in heart failure, atrial fibrillation, cancer, lung, and neurodegenerative diseases. So Blair, we like our CAM kinase, but we also maybe need to modulate our CAM kinase as we get older so that you can survive longer.
I gotta live forever.
Yes. But it's one more molecule for the researchers to look at to see if we can live longer. And they did discover that in mice and fruit flies that they messed around where they messed around with the CAM kinase 2 levels in these animals, they were able to shorten the length of the animals and insects by increasing the CAM kinase 2 activity. So when it's more active, they died younger. And then when they decreased the activation of the CAM kinase, the flies and the mice weren't able to climb as quickly. They weren't able to get food as quickly and get all the energy from it in the same way, but they lived a little bit longer as well. So target, potentially, but we need it because it's good. It's in our muscles. It's important. So how do we manage something that is really important to us and our survival, but that has a trade-off?
So, does that mean, like, exercise is bad?
Yes, Justin. No, not at all.
Bad. Buffet's good.
Yeah, exercise. This is what we're learning.
Yeah.
Thank you for joining us for TWIS. There we go.
The science is getting really, I'm starting to really like the advice.
Drink your coffee, eat your steak.
Yeah,
great. This is This Week in Science, everyone. Thank you for joining us for another episode of Science Fun. We hope that you are enjoying it and having fun. And if you are, please tell a friend. We're going to come on back right now with a little part of the show that we know and love to call Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh, I have a great story about garbage birds.
What?
Garbage birds, a.k.a. cockatoos.
What?
Sulfur-crested cockatoos. Why am I calling them garbage birds?
Yeah, they're really pretty. What are you thinking?
So pretty. They're so smart. But they belong in the garbage, or at least that's what they think. So let me tell you why. In recent years, cockatoos living in Sydney suburbs have figured out how to open household garbage cans, and they love it. They can eat all of the sandwiches, fish bones, fruit, garbage wrappers, whatever they want. They get to eat all of it. And it's so much easier than foraging for food, breaking open nuts. Definitely preferred. And so this kind of, as far as they can tell, is just some of these cockatoos in Sydney, in the suburbs, figured out how to open garbage cans. All of a sudden, the behavior started to spread. Birds in different locations.
Smart.
Yes. Started to open garbage cans all over Australia. Well, not exactly over all Australia. I'll tell you more about that in a minute. But a bunch more areas. Cockatoos opening garbage cans. And so a behavioral ecologist, Barbara Klump, at the Max Planck Institute of Animal Behavior, decided, I want to study this. Why? Because this is an amazing opportunity to study a behavior from its origin. When we think about animal behavior, we can't travel back in time and figure out the first time that a cockatoo figured out how to open a nut, how to crack open a nut, right? We can't figure out the first time a New Caledonian crow figured out to use a stick to get grubs out of a hole in a tree. But now we have a very small group of birds that know how to open trash cans, and that behavior is currently spreading. So this is an amazing opportunity to see how behaviors spread in the animal kingdom. So what they did is through social media and a mailing list from organizations like the Royal Botanic Garden and other organizations that would be interested in the environment and nature and animals, they ran public surveys. So this is a citizen science project, essentially. This is in 2018 and 2019. And they asked if people had seen this behavior, if so, when and where, how they were opening the trash cans, all this kind of stuff. Over 1,300 people responded. And then they were able to make a map to kind of see what was happening with these garbage birds. I'm going to keep calling them that because they've earned the title. And so before 2018, cockatoo dumpster diving had only been reported in 3 suburbs. By late 2019, 44 suburbs out of the nearly 500 in the survey. And so that's a substantial growth. There's still a lot of space to go. So there's an expectation that this is going to continue to spread. And the spread had a pattern. It started near the 3 original suburbs and trailed off as they got farther and farther away from those suburbs. So very clearly, these cockatoos are learning from each other in these 3 original areas. And then other cockatoos are learning from them, or learning from them, or learning from them. And it trails off as it kind of gets more and more diluted. In one neighborhood, it appeared to pop up on its own, but come on, birds can fly. So who knows, right? But that does mean that would be at least a new nexus point, right? That would be a new starting point for cockatoos to break open trash cans. And so either they hit on it independently, which is what the researchers suggest, or, as I might suggest, maybe perhaps a strong gust of wind brought a bird into a new area. But regardless, this is a new area for the garbage hunting to begin. There's also some anecdotal reports of it happening elsewhere in Australia. But anyway, this is like a very rich area to potentially study. And so they then secondarily caught and marked 486 birds in garbage can opening hotspots, and then they filmed them. They filmed 160 successful dumpster dives, wow. And they noted some common steps. So first, they would lift the can's lid at the front corner with their bill. Then they held it slightly open, and they'd waddle towards the hinges. And then finally, they would flip up the lid suddenly so that it fell open and yielded the treasure
bounty?
Trove that was the sweet bounty of the garbage buffet. See, it's all back to buffets. And so what's interesting, though, is that different individuals had different techniques. Some held the handle of the lid, others held the lid itself, some held it with bill and foot, others just used their bill. And the farther apart the dives were geographically, the more their techniques differed. So not only is just like, oh, garbage, that's a good idea. No, this is indicating that it's not just the knowledge that there's good stuff in the dumpster, but that it's actually the how is what's spreading.
Yeah.
So they call this
and
regional
it's spreading and then
subculture.
having slight mutation to the method.
Yes, exactly. Yeah. And so, so regional subculture. So this is like we've talked about with dolphins, they use sponges to forage. And so, yes, so this is the idea that this is another situation where there is what you could consider animal culture developing based on foraging techniques. And so it's not new to think that animals might figure this out. And we know that parrots and citizens in general are extremely smart. So that's not too much of a surprise. But what's really cool is to be able to see this behavior spread and differentiate across Australia. And I hope that they keep up with this research because, like I said before, this is like a golden opportunity. So rarely do you get to watch a behavior begin and spread throughout a community.
Yeah. It's the beginning of a new behavior. I wonder if other birds that are generalists, like these sulfur-crested cockatoos that come into contact with human civilization. So crows, ravens are very common. Pigeons, I'm not really going to go very far with that one. But a lot of the big-brained birds that are generalists and in contact with human civilization, I wonder how many others are paying attention and might pick it up as well.
Yeah,
Absolutely.
absolutely.
The interesting thing is to think about if humans exist for another 10,000 years, 20,000 years, 30,000 years, and we continue to have the same garbage can design.
Yes.
Yeah.
You can imagine like a bump or a little pad starting to form on the beak that's being used to sort of lift the... You know, this is how evolution starts. And what's sort of interesting then, it's not just a random maybe mutation that's taking place. It's an activity. It's a pursuit of a certain type of food over and over and over and over again that then leads to those who are also pursuing it, who have a little bit better better ability to flip open the lid, getting more of their genes out into the thing. It'd be fun to watch
I'm sure, though, that these cockatoos are creating a big mess. They're probably pulling stuff out of the garbage and throwing it all around. And so people are probably getting annoyed. And so what we're going to see is adaptation of the people. And people are going to start making it harder. They're going to put latches and locks on the garbage cans, maybe make it a little harder for the cockatoos to get access to. And then the cockatoos, will they adapt in response
come back
to this
with
energy source?
And bolt cutters.
It's an experiment in action.
Yeah, this is one of the things I was thinking about reading this story was like, oh man, it's very clear that Australia does not have raccoons because you could not have trash cans this easy to open in the United States.
It's funny you say that because literally this morning there was a raccoon out on my eating on the fence, eating berries out of the neighbor's tree.
Yeah.
Eating berries out of a tree, much cleaner than dumping over your trash.
Is that what they're supposed to? Because it seemed like they're like, maybe they're like, oh yeah, the birds are in the trash now, so we're gonna have to go back to berries.
Mm-hmm. You got another story there, Blair?
Yes, I do. It's a quickie but a goodie. So this is just it's a very convoluted story, but basically just something that I wanted to bring up for us to talk about for a second. So. This is a report on the fact that Save the Chimps, a sanctuary in Fort Pierce, Florida, has made history by allowing chimps to make their own NFTs.
They did the programming?
No. No. So actually, Save the Chimps has been working for a long time with chimp artwork. They really like to paint supplies. Paintbrushes, paint, and canvas are really common enrichment items for lots of animals, especially primates. It's definitely something that they enjoy to do, but Save the Chimps has used that as a fundraising opportunity. And now this is the first animal artwork, as far as I know, and this publication knows, of animal artwork as an NFT. So non-fungible tokens, which I could try to explain, but I don't fully understand what they are anyway. They're made up digital things that no one else can own and are therefore very expensive and are kept on a server. That's kind of what I understand. So anyway, really,
I like your explanation, Blair.
It's not completely inaccurate, right? So anyway, the especially funny thing about this, without me getting all into a tizzy about NFTs, is that the chimps physically painted paintings. And then they scanned them into the internet and made them into NFTs. So you could buy the paint, but instead people are paying for the NFTs. But regardless, the reason that this was brought up in the scientific community this week is that this reopens the long and complex debate and history of non-human animals and the art they create. Who owns it? Who benefits from it? Where does the money go? Is it actually art? Lots of questions. The thing it reminded me of was the famous selfie of a monkey, right?
Right, where the monkey picked up the photographer's camera, took its own picture, probably on accident because it's just picking up, but it was a picture, and thus, does the monkey own it? The monkey took the picture. Whose copyright is it?
Yeah. Which,
it was smiling and it wasn't focused like perfectly. So
Truth.
I don't know.
It
was
so
very nice, but-
a natural. The
thing
monkey's
is,
a natural.
if animals don't have the ability to spend the money, I don't quite but it's...
Should we treat the animals like children? Where, children are managed by their parents for acting, or, now there hopefully are some new laws coming into play for the social media platforms like TikTok and Instagram, where kids are being used for financial reasons. But same thing. I mean, if there's an organization that wants to raise money to save or help conserve certain species of animals, is that? I mean, why not? What are the monkeys going to do with their art? Do they get angry when their art is taken away?
No,
they don't care.
No,
they made their art.
no.
They had fun.
They care more about the paintbrushes themselves being taken away, because it's about the process, which is really what art's truly about, right? Is the process.
As you would know as an artist, Blair. Yes.
Hey, but isn't also part of it then what we're talking about is like we would need to convince the chimps to use money and to buy into that system. Yes. I just think it's an unconvincing argument.
Yeah, money. I agree. I think that
So is this backed up by a stash of bananas somewhere? No, not at all.
No.
Yeah.
Yes, so you own the picture of the banana. And no one else can own the picture of the banana. Do you see?
Okay. So that's I sell those.
It gives you a unique ownership.
Yeah.
Yes.
So the chimps are selling a digital image of a painting that they made.
Yes.
Yeah, I know. Okay, I know.
But there's one and only that digital image, because it has been coded and linked to this
look at
sale.
Clay's work. That looks like a brain.
There's only one.
Wow.
There are not multiple
You're saying
copies.
There's only one, but I see it right there. I can take a screenshot of it.
But that's not the one.
This is my problem. Regardless, the reason I brought it.
It's only $25.
Rag
that
on
actually
NFTs.
looks like a deal. Oh, I like that. That one by Clay.
I have
looks
a picture.
Like a brain.
I have a picture of the Mona Lisa. I mean, I took a picture of the Mona Lisa at the Louvre once upon a time.
Yeah.
So I own the Mona Lisa.
Yeah, but the difference is if you actually own it, you can touch it and hold it, and it's the resolution's better. It's whatever. NFTs, I think they're dumb. Come at me. Regardless,
it's helping. If organizations can use it as a way to fundraise and help their cause, is this a bad thing? I would love to hear from our audience. And hear what others think about this topic.
Yeah, and I think ultimately that is the key, Kiki, exactly what you said. If somebody is benefiting from the artwork of an animal, that animal should be benefiting in some way. So the thing that they came down to when they settled on the monkey selfie was a percentage of the profits went to conservation research for the rainforest or whatever. And so for these guys, these chimps are directly benefiting from this fundraising because it is fundraising for the place that they live. These are all rescue chimps that were either used in medical testing or were in the entertainment industry. And so they cannot be released into the wild. And so if the animal's benefiting from it, I think that's great. I think that similarly, if you're just taking a picture of stuff, or in the case of the photographer who's the monkey took the picture, quote unquote, it's good form. It's good form to make sure if you are benefiting from wildlife, that wildlife benefits.
I think the quotient here is the fact that these are celebrity chimps. Basically, who are being put to the forefront of the center as a fundraiser. It's not. I don't think we're in the point where we're going to have secret warehouse of deprived chimps pumping out artwork because people can't get enough chimp paintings in their lives. I don't think we have to be worried about this at all.
At this point, I do not. Yeah, I agree.
Yeah,
it's
absolutely.
Yeah, it's fine.
Yeah. You can buy an NFT painted by a chimp, or you can adopt a manatee I mean, there are all sorts of ways that you can help animals. If that is a cause that you are interested in, there are many species around the world that can use our help. On that, Blair, thank you for bringing up the interesting question of who owns it? Who gets to benefit from it? And once again, we'd love to hear from you. This is This Week in Science. Thank you for joining us for this episode. If you appreciate us coming to you every single week, please head over to TWIS.org and click on the Patreon link. Once at Patreon, you can choose your level of support and receive gifts from us in return for your support. But, I mean, really, you keep getting TWIS every week when you support us. Choose your level, $10 and up. We will thank you by name at the end of the show. And I do hope that you head on over to twist.org and click on that Patreon link right now. Thank you for your support. Can't do this without you. All right, everyone. I have a couple of fun brain stories for you. I have brainy news, as I like to bring it. First, I want to talk about parenting in birds. We know that in mammals, there are certain compounds that are released that lead to parental behavior and parental care. Now, not all birds, reptiles, not all species care for their young in the way that we think of as parental care, like really doting on the young and taking care of them. But beyond this, there are changes that can be induced by being sensitized in the presence of young. Those virgin individuals can be induced to have changes through transcriptomic ways, where there are release of certain factors in your brain that allow different hormones to be released that change the way you respond to young individuals of your species. This happens in humans, this happens in mice, it happens in all sorts of mammals, but we really didn't know whether or not it happened in birds. And so, published in Nature Scientific Reports this week, a couple of researchers, Patricia Lopez and Robert de Bruyn, they wanted to look into this question in birds, and so they took a look at a domesticated species, Japanese quail. And the Japanese quail have been domesticated to the point that they have lost their ability to care for their young. They just lay eggs and they don't pay attention to the legs. Don't pay attention to the eggs anymore because it's been bred out of them. However, when the Japanese quail are put into the presence of little baby quail, they start to have parental behaviors after a night spent with the young. And so these researchers wanted to know what happened, what's going on in the bird's brain. And they found that through sensitization, just basically getting a virgin bird together with a bunch of young baby birds overnight, that they had gene expression changes in the hypothalamus and what's called the bed nucleus of the stria terminalis. And one gene that was identified that was of interest to them is neurotensin. This is involved in maternal care in mammals. And the other is urocortin 3. And this is causally demonstrated from their abstract here, I'm quoting, to affect young-directed neglect and aggression in animals. And so their studies, they think what they're concluding from this is that it reflects core neural changes that can be associated with avian young-directed care independently of extensive hormonal stimulation. And it opens new avenues of research into understanding the neural basis of parental care in non-placental species. So it's just a fascinating story of how animals can come to be interested in taking care of their young or uninterested in taking care of their young, and why and when that happens. I just think it's really amazing that we bred it out of these birds, but then if we put them together with baby birds, they're like, oh, my brain is changing and now I must care for them. I will take care of the babies. And then they do.
You can't deny the little baby.
Right? But this, I wonder how involved this is in, so these were not the parents, right, of the little baby birds, right? So these are just, like I said, virgin birds being put together with the babies. Is this are these some of the neural changes that take place when adoption happens? So when we have, we've seen various species take on the young of others in their social group when the parents have died. And I wonder if these are the kinds of changes, if these are the kinds of neurotransmitters that start becoming active and allowing animals that are not parents to suddenly start acting like parents.
Yeah, I mean, that's also, this is a jump, but you know how domestic animals, the babies are extra cute, and we always say that's to like spark parental care, essentially, in us, most likely. But yeah, I wonder if there's something similar going on when you adopt a puppy or something, if it rewires your brain to care for this thing and to feel a certain way about it.
Yeah, and so many people do talk about them as being,
My
your fur babies. Your fur baby, yeah. And yeah, are these the kinds of, do you have a shift in your brain that's like, this is, you're going to take care of a baby now. And you need to have these things active so that you're not aggressive and so that you're caring and so that you're giving them what they need.
You have an emotional response when they act distressed.
Mm-hmm.
Yeah.
Oh, there's something wrong. I have to fix you. What can I do? Yes. What else do we have? Also, in the brainy news, I also have some sleepy
How dare
news.
you? How dare you?
And it's not about yawning, but it is to do with our need to sleep. So we all know sleep is important, right? You need to sleep. When you don't sleep, things start just going wrong, right? If you're not sleeping, suddenly you're like, you start forgetting things, you're clumsy, like just things start going downhill pretty quickly.
You're describing my day today. Yes.
What these researchers at the University of Michigan have discovered and published in their latest study is that in the Proceedings of the National Academy of Sciences have found that there is an actual shift in your brain when you do not sleep. They looked at neurons in the hippocampus. The hippocampus is an area of the brain responsible for navigation and also very important for consolidating your memories, taking in information, linking it all together and going, okay, we got to store this stuff somewhere in the brain. Okay, the hippocampus is active to help you remember things. All right, so they looked into the brain and looked at the interaction in hippocampal neuron activity, how things changed between sleeping and waking, and specifically at what's called phosphorylation of the S6 component of ribosomes. The ribosomes are little tiny organelles that help turn mRNA into proteins. So you can imagine, very important for protein, the creation of proteins that are going to be involved in memories and memory formation. They used mice for this study, and of course, because learning in mice, it's all about fear, they used a fear stimulus. And when the mice were allowed to fall asleep following the stimulus, they saw that this S6 phosphorylation increased in the dentate gyrus portion of the hippocampus. And this is the first part of the hippocampus where memory information starts to come in. Without sleep, what do you think happened to phosphorylation in S6? Decreased all through the hippocampus. It was down. And the fear memory was disrupted. They did not remember the event, that the fear stimulus, as well as the individual mice that were allowed to sleep. So they wanted to know exactly what was going on there. They found that this whole process, there was increase in abundance of RNA transcripts that are present in interneurons expressing a neuropeptide called somatostatin, as well as an inhibitory neurotransmitter called GABA. The interneurons with GABA, if they're releasing that, then they are inhibiting surrounding neurons and inhibiting all that phosphorylation in the dentate gyrus in the hippocampus, slowing down firing, making less activity happen. And this is all happening because you are not sleeping. You need to go to sleep.
You have to turn off your computer every once in a while. It needs a restart, right? Your brain needs it. Your brain needs to turn off.
Yeah, and if it doesn't get to turn off, it starts telling itself to turn off. It's like, I'm going to put the slow down switch on. It's like, when you don't get sleep, it's like, inhibit. Why are you not asleep yet? Inhibit. I don't want to remember things. I should be sleeping. Your brain is trying to tell you something. Yes. I mean, I might be anthropomorphizing your brain just a little bit.
Just a little.
No, I feel like this is the internal conversation. I had 3 hours, I think, of sleep last night, which for me, for some people, they can function. No, it was not enough. It was very bad. And I had to, yeah. So I'm going to, as soon as we're done, I'm going to go to sleep.
I hope you do get a very good night's sleep so that you will have plenty of S6 phosphorylation in your hippocampus.
I didn't even know I was missing it, and that was the problem. And now I know why I was tired this morning and couldn't remember anything. I didn't have enough S6 stuff.
Yeah, no S6 phosphorylation. You don't have any of those memory transfer proteins. No memories, or less memories anyway. Memory formation inhibited in your hippocampus when you don't sleep. So, Justin, it's your turn to tell us a couple of stories.
All right.
Do you remember what you wanted to talk about? No, I'm kidding.
Yeah, there was 250 million years ago. Much of the life on the planet was dying.
No.
It was an event that marked the end of the Permian period. 96% of the planet's marine species, 70% of land life, went extinct in a relatively short period of time. Largest extinction event in the history of the planet. Florida State University researchers, they looked at this. They published in Nature Geoscience. They found actually that there was a sudden spike and then subsequent drop in the ocean's oxygen content. Now, the drop in the oxygen content had sort of been known, but the spike of oxygen in the beginning of it was a new finding. One of the things they say is that although ancient marine oxygen levels were on a downward trend ahead of the spike and remained low afterwards, it's on a very geologically rapid shift back and forth. They think it happened over just a course of about 10,000 years, something like this. So it's a very fast event that took place that changed oxygen levels. And sort of like you were describing that deep pond thing where like the sunlight
the sinkhole.
Get in, the sinkhole, where you could sort of see the alternating population shifts during daytime and nighttime. You know, these massive shifts of oxygenation in the ocean may have been a big key driver to the mass extinction event. One of the other things is they also know that there was a lot of volcanic activity. So the lower oxygen levels, and then you have the only reason I think is what you were talking about is the massive volcanic activity could have been a dimming event for sunlight making it.
Oh, absolutely.
Right. So then there's a whole other reason this
component.
Yeah. So anyway, interesting. Oxygen. It's what we all breathe and keeps us alive, but it's also important for everything now on the planet. This is an older extinction event where, you were talking about the red-green algae were producing oxygen when most things probably were more like that sulfur eater. Like, there was a time when oxygen was a toxin to life on the planet. And the red-green algae producing so much oxygen, I think, actually led to an extinction event there for a lot of the critters that didn't like it. Also in the news here, glaciers and ice caps in 2 archipelagos of the Russian Arctic are losing, I like this, enough meltwater to fill nearly 5 million Olympic-sized swimming pools each year. Which is a scientific quantification, by the way. That's how they talk about large volumes of water as units of Olympic-sized swimming pools. Satellite data suggests that the amount of ice lost between 2010 and 2018 would put an area the size of the Netherlands 7 feet underwater. They didn't do it in Rhode Island, which is unfortunate. That's an American. That's how we usually do things in units of Rhode Island, not units of Netherlands. But okay. Warming of the Arctic Ocean appears to play a key role in accelerating ice loss from 2 groups of islands that border the Kara Sea. Researchers said they rely quite heavily on satellite data, mostly because these are areas that are hard to access. They're up in that Arctic region. It's really cold, and there's not a whole lot of support services. But yeah, they say thinning of the ice has already had a major impact on the stability of some of the region's glaciers and ice caps, which could further increase ice loss in the future.
Apparently, there would be around 16 Rhode Islands in the Netherlands. So, the Netherlands
16 Rhode Islands. See, now I can conceptualize
15 to 16 Rhode Islands in
now
the Netherlands.
I can actually conceptualize that under 7 feet of water. 15 Rhode Islands.
Yeah,
under 7 feet of water. That's pretty impressive.
That's a lot of water.
That's a lot of water now. Yeah. That's the story. Yeah, Russia's melting. It's not just Greenland. It's not just Antarctica. It's not just the caps and the Sierras. It's everywhere. It's happening everywhere.
So this is the Russian Arctic. It's not Siberia. This isn't just the permafrost. This is frozen ice over land, similar to our Arctic, just on the other part of the planet. Yeah.
If it's ice, it's going to melt.
And if it's Arctic, it's Arctic. It doesn't matter whether it's United States Arctic or Russian Arctic. It doesn't matter. It's all the far north, and it's melting because climate is changing.
Climate change does not respect lines drawn on maps.
No, it absolutely doesn't care. And we're going to... the last few weeks, because IPCC stuff is happening, a lot of researchers are talking about climate change, and politicians are discussing what we're going to do and how we're going to address things. But we're going to have to get ready for ice-free Arctic. And Russian Arctic, American Arctic. What is going to happen there? We're seeing plastics being discovered, microplastics found in the Arctic. We are also, if shipping increases in the Arctic, we're going to see further pollution through diesel, oil, gas. If there is mining, if there is further searching for fossil fuels in the Arctic.
Which is exactly why Blair's shortening the story about shortening the legs of spiders needs to be implemented in humans, to have smaller humans, to use less resources, just to give the planet some time to recover before we do it again.
Yeah. We need a little bit of recovery. Please, everyone. Yes, well, we need critical thinking. I love your comment. Yeah, if there's fresh water coming from that, can we pipe it to California? The American West could use it. Great solution.
And then this is always the part where I have to point out that California still has plenty of fresh water for all of the people. And for the rivers.
It's just when you add in all the farming and the almonds. I'm really upset with almonds.
It's just the almonds. It's really just get rid of the almonds and we'll be fine.
Yeah, can we just, I mean, is it inaccurate to blame it on the almonds?
No,
I don't feel like
isn't.
it is.
It is not. Yeah, the whole thing when they're talking drought in California, like personal people consuming water in California, accounts for about 10 to 11% of the fresh water use. The other 90% is farming and industry.
Yeah.
So, yeah, like they're telling people not to water their lawns when they could just be getting rid of some of the almond fields that they built
while the drought was building
while the drought was
up.
already known
Yeah.
and building. They were opening more and more territory for almond growing, which this is like probably the number 2 thing I eat. Like I would go to the buffet and eat nothing but almonds if they were available. I love almonds. It's my favorite.
You're
food.
a part of the problem. I see how it is.
I am the problem. No, I'm not. I'm not saying that I'm not. I'm just saying, there's not really enough of a drought in California to be.
So I have a question as a possible solution. We are seeing artificial intelligence being used to find patterns in all sorts of things, to find solutions for protein folding, to solve complex problems in resource distribution. People are using artificial intelligence to work the stock market. We're using artificial intelligence to identify cancers better than doctors can now. Can we develop artificial intelligence for complex systems to help us distribute our water?
No.
Would that be a good solution to this problem?
So the thing is going to spit out an answer.
Yeah.
And the answer isn't going to be liked by somebody who has a tremendous amount of money. And then
for
the
sure.
answer isn't going anywhere.
I was going to say a similar thing and say that we could also ask the AI to find the solution to climate change, but it's going to be to reduce our use of fossil fuels, and no one's going to want to do that. So,
well, what I think, that we could do is that we could tie this AI in to the AI that controls the stock markets and bids in the stock markets. And potentially we could get the people with money to follow along with what the climate change and water distribution AI wants to do, because it will affect the stock markets in such a way that it will take away all their money if they don't do what the AI wants.
So there's absolutely, I 100%.
We need
100%.
a benevolent AI to rule everything.
This gets tricky.
What could go wrong?
There is always an argument for, and I 100% believe that you could change those levers of resource flow and create a system that is working better for everyone. The problem, no matter how good the system or bad the system currently is, somebody's winning. And whoever is winning does not want change. Even if it could be a little bit better for them, but maybe it would be a lot better for a lot of other people. If you're winning the system of capitalism money thing, grab, you don't
you're not
want
gonna
any...
you're not gonna like the AI telling you what to do. I get it.
Right.
Yes.
And that's who, unfortunately, holds undue power in the system of governance that we have that is fueled by that money.
This is like what we've been dealing with climate change, and now again with trying to get people vaccinated, where we're like, what if this person says it? Will you listen? No? How about what if this person? What if a robot? No? What if Paul McCartney? No? What if the no? What? So yes, I don't
Blair,
know
what is the most popular owl?
What is the superb owl? Popular how?
Who gives a hoot?
Oh.
True. I like the burrowing owl. I think burrowing owls are very adorable.
A diurnal owl, of all things.
Yes, diurnal owl.
There's the barn owl, only owl in its family, sounds like a dying banshee. Yeah. There's the great gray owl, the tallest owl. There's the Eurasian eagle owl, the heaviest owl at a whopping 5 pounds.
I think the Eurasian eagle owl is a pretty amazing owl.
I also really like the saw-whet owl, which is like this big.
Yeah.
It has that name because it sounds like you're whetting a saw.
There's one that was a little ground owl that I think was in Greece, but it was on all of their money. It was like one of the first mass-produced coins, and it's a little owl.
Well, today is Owl Appreciation Day, so I hope everyone appreciates a little owl today. And if anyone wants to send us their opinion on their favorite owl or on their whether or not you would listen to a benevolent AI, you know where to find us. Have we made it to the end of another episode?
Yes.
I think we might have.
Made it. We might have made it to the end. We might have. And I think I might have shut my show notes up. What did I do there? Doesn't matter. I want to say thank you for joining us for another episode of This Week in Science. Thank you for watching, for listening, for being a part of this adventure, this science stream. And I do want to say a very specific thank you to a few people who help out the show. Thank you to our moderators, everyone who moderates the chat a bit. You are absolutely helping us do these streams and make sure that we can maintain the community, the kind community online that we have. Thank you for doing that. Thank you, Fada, for help with show notes and show descriptions. Gordon McLeod, thank you for your help in the chat rooms. Identity 4, thank you for recording the show. And Rachel, thank you for your assistance now in editing. It's very wonderful to get that help. And I definitely want to thank our Patreon sponsors. Thank you to Pierre Velazarb, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Melanie Stegman, DeCramsta, Karen Tozzi, Woody MS, Andre Bissett, Chris Wozniak, Dave Bunn, Vegard Shefstead, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Shubru, Don Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredis104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Hey Arizona, Support Aaron, Lieberman for Governor, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Mallory Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DePel, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if you are interested in supporting us on Patreon, head over to twist.org and click the Patreon link. On next week's show,
we will be back Wednesday, 8 p.m. Pacific Time, broadcasting live from our YouTube and Facebook channels and from twist.org/live.
Hey, do you want to listen to us as a podcast? Maybe get in some multitasking, go for a nice run, do some dishes while you listen. Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, you can get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to the stories will be available on our website, www.twist.org, and you can sign up for our newsletter.
You can also contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in that subject line, or your email will be spam filtered into the mouth of a snowy owl. It will be partially digested in their gizzard, and then it will be coughed back up in the form of an owl pellet, and we still won't see it.
Hey, if that doesn't sound appeasing, you can just hit us up on the Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We'll love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views. But I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought. And I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science.